there are still an abundance of studies which show different benefits of a ketogenic diet. But how can it be beneficial if it actively suppresses bifidobacteria, which are considered the really good guys? These are not necessarily the really good guys. It depends on context. Again, in a ketogenic context, they are not even not needed. They are actively suppressed by the ketones.
Welcome to the Recovery and Transformation Podcast, the show that links personal health with societal well-being. I'm your host, Samir Dosani. I'm an activist, a PhD student, and a health coach based out of Johannesburg, South Africa. This show explores the root causes of disease and talks about how people are recovering and transforming every day. Hi, and welcome to another episode. My guest today is Gabor Erdosi. Gabor is an obsessive study reader and analyzer, fascinated by everything related to metabolic disease. He's recently started delving into small intestinal interactions between foods, microbes, and host physiology/immunity. He's discovered that this is the place where everything starts, the true root cause of most diseases of modernity. In this discussion, Gabor and I talk about the microbiome and some myths around the microbiome. Gabor goes kind of deep into a couple of studies, some recent, some not so recent, on what the microbiome needs to thrive and what we know about it. Also some interesting animal models. In general, it's a great discussion. I hope you enjoy it. Gabor Erdosi, it's a real pleasure to have you on the show.
Yeah, thanks a lot, Samir, for inviting me.
So Gabor, I got to know your story with a great interview you had in a pub somewhere in Europe. I forget where. Was it in Hungary or Ireland or somewhere else with Ivor many years ago? But for those who don't know, please just tell us a little bit about your background and how you came to do this work on metabolism.
Yeah, first of all, I have to apologize because we adopted a parrot and it might be audibly disturbing to hear. He really loves my daughter, and when she's not at home, he tends to disturb me like crazy and shouting like crazy. So sorry for that. Yeah, I think that interview took place something like 6 years ago with Ivor Cummins. He was visiting Hungary at that time, and he invited me for lunch, and I think we recorded the interview then. At that time, I think I was already turning towards the gut as the most likely source of the vast majority of modern diseases of civilization. And I was really fascinated by the kind of imbalanced responses to highly processed foods, what you can measure when you follow gut hormones, hormonal responses to different food items. And yeah, I think we were mainly discussing that. Regarding my background, originally I'm a molecular biologist who graduated in the mid-90s, and then I did an MBA out of interest in moving ahead in corporate business. And yeah, I was doing different things through my career as an employee first. I worked in the medical devices industry, sold different I was in product management and sales of different devices used in surgical procedures. So I attended a lot of surgeries live and even advised surgeons in some of the aspects. And yeah, among that, there was one interesting thing gaining traction back at that time, and it was bariatric surgery. And it was a really interesting experience, or yeah, professional experience, which I could link to the gut physiology issues later. Then I moved on to industrial biotech. I worked with enzymes and microorganisms in the mainly in the so-called grain processing industries. So starch breakdown, also to some extent sugar crushing and producing sugars and producing high fructose and another types of syrups. And what else? Then I went and worked for a syrup producer, a local, newly built plant. I had come online. Then also worked as a specialist who supported the sales department. So we went out and visited different food industry customers who use the different syrups. So I'm also familiar with all the use of all the processed food industry stuff, how you use the sugars, the syrups, the starches, the modified starches, maltodextrins, and all these kind of things. And most recently, I've been working as a consultant, mostly for smaller companies and startups. Sometimes I also do individual consulting, but I'm not really the usual eat this, eat that, and move more type of health consultant. I'm more into the scientific details, so to say. So usually people who come to me already tried a couple of approaches and failed a couple of diets and tried to get another perspective on what may be happening to them. And I think I recently I've been working recently on three projects. There is a health app project, which tries to model all modern diseases with a Bayesian network. So it will be an app which grabs all the information provided by the people and lab data, and then tries to identify the problem at hand and also provide automatic recommendations. So, and that's fairly heavily on a scientific basis. So we have been building the scientific core of this app for 1.5 years already, and I think there is at least one more year to come to the acceptable automatic solution. I also work with another project called Metabolic, who try to bring insulin measurements mostly, but it's all kinds of metabolic measurements to your home and to the GP's office, so that you don't need to go to a hospital to get your mostly dynamic glucose, triglycerides, and insulin measurements. So you can have a better picture of your metabolic health by using their tools. And yeah, for more than a year, I have had this joint venture with Ivor Cummins called Metabolic Duo, where we have bi-weekly sessions with interested people where we discuss mostly metabolism related stuff, but also some other biology, which can be of interest
Yeah.
for people.
Fascinating stuff. Fascinating stuff, Gabor. So what I wanted to talk with you about today specifically is this idea. So there's a hypothesis being thrown about. It's being thrown about rather loosely, I can say, by people like Chris Gardner, sort of the pro-vegan kind of doctors, other people, that Zoe network in the UK as well, many of them, who say, look, when I say, so me as someone who advocates more carnivorous kind of a diet, I will say, look, humans don't eat fiber, humans can't digest fiber. But they said, no, you're right. Humans cannot digest fiber, but the bugs in our gut, they really need the fiber. So that led me to ask this question: is it true that the bugs in our gut need the fiber? And if so, what's the evidence for that, and what's the evidence against that? So with that, maybe I'll turn it over to you to share some. I believe you have some slides you can share with us, or some further thoughts.
Regarding your question, if I wanted to answer it with a single sentence, I would say that yes, the microbe also need food or feed. And that doesn't necessarily mean that you as a host needs to consume that fiber for the microbes. So it may sound controversial, but actually it makes a lot of sense if you start thinking about scenarios where you do not consume any fiber or very, very limited amounts of fiber, but these scenarios are still considered as healthy lifestyle choices or evolutionarily appropriate lifestyle choices or ancestral types of choices. And in this presentation, I brought together a couple of examples. First of all, there is the claim that your microbes will eat you alive if you don't provide them with the exogenous or external plant fiber. That's usually what people mean by fiber, that you need to eat all those fibrous plant stuffs, which you cannot digest, but your microbes absolutely require. And yeah, I'm going to show why that is probably not a proper interpretation of the evidence. And also bring the examples of fasting, breastfeeding, and the not necessarily human example of carnivores, because these examples show that you don't really need to consume the plant fibers to stay healthy or even become healthy. Obviously, when you fast, you consume nothing, including fiber. So no fiber, no protein, no fat, nothing, because you are fasting. And the longest, I think, medically documented fast is something like 390 days, so more than a year without any fiber or any food. This gentleman only consumed liquids and some vitamin pills to be on the safe side. And when you think about it, an infant can be breastfed for a year, exclusively breastfed for a year or even more. And that food doesn't contain any plant fiber, if I'm not mistaken. And in carnivores, there is often that claim that carnivores also eat the bowel content of various animals and sometimes chew on grass and these kind of things. But I think it's just an overshoot that they consume those things, when they do that at all, for the fiber content in those foodstuffs. So where these microbes eat you alive message originates from, I think that the prime suspect is a study from 2016, and it was published in the journal, in the prestigious journal called Cell. And you can see the title in this bluish clip. A dietary fiber-deprived gut microbiota degrades the colonic mucus barrier and enhances pathogen susceptibility. What does that mean? That essentially means that if you deprive your gut microbes from fiber, then the mucus barrier, the gelatinous layer that covers your intestinal tract from the inside, and also other barrier sites, will be degraded by the bacteria, and then pathogens will thrive. And if you have a look at this study, I think two things are very obvious: that it's a mouse study using an ultra-refined lab diet, which is often called a high-fat diet. And a high-fat diet consists of lard, typically lard, or what else? Canola oil or soybean oil or some oil, and some single protein source, probably from dairy protein, casein, I think, the main protein they use, isolated. And then they also had sugar and maltodextrins. So it's something like 40%, 45% typically calories come from fats by energy, and another 40% comes from carbohydrates like sugar and maltodextrins, and something like 15% protein. But I think that cannot be really considered a proper human diet, and not even a mouse diet, because it's totally ultra refined. It's uh, composed of various ultra refined ingredients. And then there is another study on the right hand side in this slide where it was shown that when mice totally lack mucus due to genetic, I think it's an intentionally generated genetic defect, they cannot produce any mucus, then they are protected from fatty liver disease and obesity. Typically, when you feed mice with this so-called high-fat diet, they develop obesity, fatty liver disease, diabetes-like symptoms. So it's a quick model of creating something similar that happens in humans. At least most researchers believe that it is similar. And what happens when they don't even have this mucus lining their intestinal tract, that they don't develop fatty liver disease and obesity? I think that's kind of contradictory to the first example. And then what may be the reason? The review, the third one in this slide, the mucus and mucins of the goblet cells and enterocytes are the regular cells in the intestinal tract. They absorb different things. And the goblet cells are those which produce the mucus. So these provide the first defense line of the gastrointestinal tract and interact with the immune system. I think that's a very important point and provides a necessary context to the others. That the context is that when there is something which the animal or the human hasn't encountered yet, it's totally not adapted to. Only the adapted adaptation repertoire of defense can be launched. So when you see a new type of so-called dyshomeostatic, so when it changes the balance in the body, this environmental impact, you have a kind of a tool set which you have adapted to use. And if it's a novel type of impact, you can only select from a tool set what you already have. And sometimes the response cannot be the right one because it's a totally new type of development. And I think these ultra refined diets, for example, are totally new. They hadn't existed until something like 50 years ago. And that's probably one of the reasons why we cannot react properly to these diets. And you need to know, you also need to know that the largest chunk of the immune system is located in the gut. So the gut faces trillions of bacteria, but also other microbes directly in the gut, and these microbes have to be kept under control, and there's an um, evolutionary way of how we deal with it and how most animals deal with it. I say most because, quite interestingly, as a side note, some animals don't seem to have a gut microbiota, especially some insects and other types of animals. But it's almost ubiquitous. So most, I think that the vast majority or all mammals have a fixed microbiota, somewhat plastic, but otherwise also quite fixed how it works. And then you need to think about it how this three-way relationship has evolved over hundreds of thousands or millions of years, because it started way before humans appeared on the scene. And the food consumed, the reaction of the host and the immune system, this interaction is very poorly understood, in my opinion, and that's why we hear all these claims of what happens with your microbes and what and you can very often hear that your microbes control this process in the body, your microbes control that process in your body, as if there was no control by the host, by yourself at all. Everything is controlled by the microbes. I think it's at least a two-way interaction, and then it's not only up to the microbes, but it's an interaction between your immune system and the microbes in the gut and the food, which is a great influencer of microbiome composition. That's known. And the first example, what happens when you are fasting? So you don't eat anything for at least half a day, but if you do extended fasting, which is more interesting here, then it can be days or even weeks or months of fasting. So and days of multi-day fasting is generally considered as not only safe but as a health promoting intervention or procedure. And how this sounds very controversial, that if you don't provide food for your microbes, then they should be eating you away by small chunks, and in several weeks nothing remains but a body chewed up from the inside. But that's actually not the case. And this study I just used as an example that when you fast, there is an increased abundance of two gut microbes, which are generally considered highly beneficial, producing short-chain fatty acids and providing the host with the necessary information that everything is okay. And quite curiously, when you stop eating, the abundance of these beneficial bugs goes up. And how is that possible? It is not absolutely not in line with what we tend to believe about feeding the bugs. And fasting intervention and its clinical effects on the human host and microbiome. And again, beneficial changes are reported. Fasting consistently enriches widely recognized anti-inflammatory gut commensals such as Faecalibacterium and other short-chain fatty acid producers. So how can they proliferate, and how can they get more abundant when you don't eat anything? What do they eat? They cannot grow without food, obviously. And then we come back to this mucus layer, this gelatinous layer, which consists of a kind of a protein network and sugar chains, but these are usually more, I call them exotic sugars. So it's not just glucose and fructose, but also fructose and xylose and other more exotic types of sugars in medium chain lengths. And these are largely non-digestible to mammals, and they are specifically there for two reasons. One is to provide this gelatinous thick structure as a physical barrier, and also to feed bacteria. So you can actually feed bacteria. And what happens during fasting was estimated. I say estimated because these bacterial measurements are still in their infancy, and we know that it's not even the species level which matters, but the strain level. If you think about E. coli, usually considered as a pathogen, but there are strains of E. coli, the same species, which work as probiotics. So it's not as easy as it seems. So you really need to dig into this and find out that which studies make more sense and which studies make less sense. And the point I clipped this image from a study, from that fasting study, is that it's impossible to tell from this enriched or depleted. So some bacteria, the abundance increases, and for other bacteria, it drops, and it's totally impossible to say that this is beneficial or not beneficial without having a much deeper understanding of this question. Yeah, what happens in carnivores? when they fast, it's a 3-day fast in farmed mink. Mink is a fairly good animal model for several reasons. If you just think about how we use them for viruses, they are surprisingly close to humans from many aspects. What happened when they fasted? The mucosa-associated bacterial community within the colon is diverse and dissimilar from the community found in the feed. So this mink study examined what happens to the microbes attached to the mucus layer of the GI tract. And what they found was that, the food largely determines the bacterial composition within the lumen, so within the pipe of the intestinal tract. But those bacteria attached to the mucus barely change when they do a 3-day fasting. That's fairly interesting, and I think it has far-reaching implications, because most of the studies that are used in humans and in animal models use fecal matter. So they use animal crap or human excrement to look at the bacterial composition, and it seems that it's absolutely not the important part to look at. And even more so if you consider that these interactions occur at the huge surface area of the GI tract, and you know that about 90% of that surface area can be found in the small intestinal tract, more specifically, and almost nobody is looking at the small intestinal mucosal bacterial interactions, then you know that something is wrong with these whole science branch. I usually use the example of learning to dance by looking the dancers through the keyhole of the ballroom. That's what we are doing when we measure fecal bacterial samples. We have just a slightest clue. It's impossible to learn to dance by just watching through the keyhole and seeing some legs moving around. To drawing the right conclusion is almost impossible. What is actually happening? So these are the main problems where all these faulty conclusions come from, in my opinion. When you fast, you are feeding the bacteria in the mucus by actually increasing mucus production. So you are not eaten alive, but your weight loss, what you experience during a fast, the weight loss comes not only from burning your fat reserves, but also providing some limited amount of food for the very important part of your bacteria, not the 2 kilograms, like, I don't know, 4 pounds of bacteria totally in your digestive system, but only to those which adhere to your the surfaces of your digestive tract. An old study I got, I don't know, way more than 10 years ago, is sorry, deals with cheetah fecal inoculum. It's not an in vivo, so it wasn't done live. The cheetah was not fed with certain foods, but rather the cheetah's microbes were harvested and used in a fermenter to try what raw materials they can utilize. And the result was actually enlightening, because the conclusion was that this study provides the first insight into the potential of animal tissues to influence large intestinal fermentation in a strict carnivore, and indicates that animal tissues have potentially similar functions as soluble or insoluble plant fibers. So now we are talking about animal fiber from the perspective that some of the animal proteins or modified proteins can serve as a substrate for bacteria to produce short-chain fatty acids. It works quite similarly to what happens to animal plant fiber. And I think the prime example of plant fiber is inulin, or more precisely, the fructo-oligosaccharides, the breakdown products of inulin, which you can find, for example, in artichokes. And there is a reason it is often called fartichoke, because it produces so much gas and large amounts of short-chain fatty acids due to the active fermentation going on. But it turned out that actually the cheetah's microbiota was able to ferment collagen, in connective tissues, as well as different animal components like chondroitin, and mostly in connective tissues, in fact. So those proteins in connective tissues can serve as kind of an animal fiber. If you don't eat any plants, but eat a nose to tail type of carnivorous diet, you most likely get the necessary feed for bacteria. And I can just corroborate that. I myself, if I consume, for example, we have these fried pork skin. I don't know the proper English term for that, but
They call them crackling, or in the Philippines, they call them
yeah, the cracklings.
yeah.
You just fry the pork skin, so it's some connective tissue and the skin itself. And I made the mistake sometimes that I consume that together with kefir, the fermented dairy product. When you ferment milk, not only by bacteria but also adding yeast. So it's a joint fermentation by yeast and bacteria. And when I had kefir together with those cracklings, the amount of bloating and gas was very, very similar to when I eat high FODMAP plant fiber type of diet, like artichokes or whatever these or beans, high fiber types of plant foods. I had typically had the typical symptoms. Really, I was bloated, and I was crazy surprised what the hell was happening, because I only had like animal foods, but it works the same. So this is, I think, another line of evidence that we need to understand the details, and in biology, the context always is always king. You need to understand the proper context, because anything derived from even sometimes from a slightly different context can have vastly different implications. It can mean something very different. And that's why some of these mouse studies, for example, using that so-called high-fat diet, are not fully relevant to some of the similarly looking human diets, which are actually not similar if you go into the details and understand how different food components work in the body. So what is the mucus? I think I already talked about it a little bit. In scientific terms, it's called a peptidoglycan. Glycan means attached sugars, so it contains sugars, and the peptido means that it has a protein core. So you have a protein network, and then, as you see in the different zoom stages of the image, and then there are these different sugars. You see the rectangle, blue rectangle, the red triangle, and then the various others. And you can see what I call exotic sugars, sialic acid, fucose, and other galactose and other sugars in a relatively short chain of sugars, but it's already indigestible to mammals. It's clearly for the bacteria. And that is key. And following the dynamics of this mucus, how they change on the different diets, and what may happen when the problem in the very first study, which claimed that you are being eaten alive, that's a different context you realize. Yeah, and then some bacteria are specifically attracted, kind of fed by these mucin glycans, and by influencing the amount and the composition of the sugars in this mucus, you can have a great influence on actually what types of bacteria thrive and what are competed out by others in your gut. So that's what basically happens when you fast. You eat nothing, you increase the mucus release, more mucus is produced, and the composition of mucus changes so that it is enriched in those sugars or short sugar chains, which the so-called beneficial bacteria are attracted to. So that's, I think, in a nutshell, it's that simple. And yeah, what happens to the intestinal ecosystem? That's the balance, the three-way balance, which which becomes a two-way balance when you don't eat. When you take out the food part, then it becomes a two-way balance, bacteria and immune system. And I highlighted some of these some of the parts. During fasting, bacteria utilizing host-derived substrates proliferate at the expense of those relying on dietary substrate. That's quite clear. So it's a dynamic change, and the masses of bacteria can die out, and they won't eat you alive because they are just excreted, and your excretions will decrease over time. If you extend your fasting from 3 days to 1 week, 2 weeks, whatever, what you experience is that your excretions diminish. And actually, something like 85% of your excretions, the fecal matter, is bacteria. And that's why it goes way down, because you stop nurturing bacteria by food. They have no food, they die out, and your intestinal content decreases. During fasting, the intestinal mucosa is renewed concomitantly to changes in immune function and the decreased inflammatory state. So if you are if you are being eaten alive, then it's most likely, or I would say with very high confidence, that it's inflammatory. So if you are being alive, then your inflammation goes up like crazy. And what happens during fasting? Your inflammation goes down. So it means that there is a different balance found between your immune system and the bacteria inhabiting your gut. And another one I highlighted, animal studies have limited relevance since rodents with a high metabolic rate can rapidly enter starvation. So it's not like they can fast easily or efficiently. They enter into starvation. They start degrading their protein stores, their muscles much more quickly than humans or larger animals do. So I think we always have to treat, and then I get back to the very first slide again, that it was a mouse study. It's not 100% translatable to what happens in humans. The diet is not identical. The model animal is not identical. You can find better model organisms when you use dogs or even the minks we discussed previously. I added another study because it's very interesting. Most people are not aware what happens when you are actually feeding. You eat some amount of protein. If you eat adequate protein, you eat something like, let's say, 20% of your calories comes from protein. And you may think that your intestinal tract is a relatively small organ if you exclude bacteria. So it may use just a tiny proportion of that protein or of the nutrients. But actually, what these scientists, researchers found used in piglets, I think pigs are a much better model organism than mice. And what they found that some of the amino acids were extracted, or like 60% was used up by the gut, and it didn't even reach the other parts of the body. So you see that 56% of the essential amino acid intake appeared in the portal blood, which means that 44% was extracted. Almost half of the protein was extracted. And what the hell happens with that protein if it's not more intestines built? It goes into the peptidoglycans and the other immune things, because we also release these different peptides which have antimicrobial effects, also to modulate, to control what bacteria can proliferate and what are being suppressed. So actually, it seems that the intestinal tract has been neglected, by to a large extent, and some of the amino acids are extracted by really the masses, by the intestinal tract, and used to feeding or fighting bacteria as the immune system sees it. And then, yeah, I think this is from the cheetah study, some more details. And what you see that it's not only the glycans which matter. We talk about the sugar chains, the glycans in connection with the mucus, and also in connection with plant fiber. But some of the proteins, like collagen, contains very, very limited amounts of glycans attached, and they can still serve as a good substrate for short-chain fatty acid production for fermentation by bacteria. You see that the highest amounts of short-chain fatty acids were produced from fructooligosaccharides. We mentioned the inulin breakdown products. Those are the fructooligosaccharides. I think that those are also the FO in the FODMAP stuff. So all things which you are reacting sometimes badly in plant food. One is the fructooligosaccharides. I wouldn't say it's a magic stuff if you become bloated from that, because it's not only the short-chain fatty acid, but some of the gas also was high from the. And then what you can see that that collagen provided high amounts, glucosamine, animal peptidoglycan also provided high amounts of short-chain fatty acids, and even casein provided relatively high amounts of short-chain fatty acids, but the distribution between different short-chain fatty acids can vary. So I would say that usually the connective tissue derived proteins are good for bacterial fermentation. Yeah, another interesting study. This is now not about depriving bacteria from feed, but providing different protein types of feed, because it turns out that these proteins can also serve as feed or fermentation raw material for bacteria. And this was a study done, I think, on rats. But the interesting thing was that they used 5 different types of animal proteins, and they also used soy protein. And this LPS, what was measured, the LBP, the serum LBP, LBP means lipopolysaccharide, that is LPS, lipopolysaccharide binding protein. So the LBP is short for that. Lipopolysaccharide is a bacterial cell wall component, and some of these LPS molecules are very highly inflammatory. They are implicated in different diseases when the amount of production increases in the gut, and also more can somehow get through the gut wall. And that this is quite an astonishing difference, how much of this LBP, which is a marker of LPS bound in the body, increased on soy and was relatively suppressed on different other types of animal proteins. So it's not only about the sugars, it's also about the proteins. It's important to remember. And then another quite different context, the breastfed human or any other mammal for that matter. What happens? How do they get the fiber? The infants of mammals are not really good mucus producers. They don't have the immune system matured closely after birth, so they don't produce. They have a very thin line of mucus, so defenses are down, and they don't eat plant fibers either. But what happens with their gut? Are they being eaten alive? No, absolutely not. The case is that they are being fed with, again, animal fiber from the mother. And this animal fiber we call human milk oligosaccharides or HMOs. These are sugar chains specifically formulated to feed, again, some types of bacteria. And the infant starts increasing their bifidobacteria, the infant microbiome is dominated by bifidobacteria. That's not the case in adults. That's a typical infant or toddler type of microbiota. And the milk specifically feeds those bacteria, which is quite unsurprising. But again, you see the chains over there. And actually, some of the lactose itself, we often talk about the high carbohydrate nature of breast milk. I don't know how much percentage it's sugars and carbs, but actually much of that carbs, all the oligosaccharides, and some of the lactose goes in to feed the bacteria, and it's not absorbed by the infant. So it's not actually that high carbohydrate than some people like to claim. But again, it's an example how we can solve this problem with ancestral adaptation, ways of adaptations. You don't necessarily need plant fiber. And actually, if you think about it, it's probably much easier to control the bacterial composition when you have no third factor to deal with. Because what comes in your food, you have no perfect control of. You even if you calculate meticulously that I am eating this much amount of this type of fiber and that much amount of another type of fiber, it's very inaccurate. And also you don't really know what type of bacteria are in your gut, in what numbers. So you don't really know how to feed them the best way. So I think the best way to control them, if you have any gut-related issues, and most modern diseases are gut-related issues, the best way is to eliminate the unnecessary third factor, the food factor, and do a multi-day fast, go on a low connective tissue carnivore diet first. I call this an elimination diet. Like fasting is also an elimination diet, but you cannot fast, especially if you are lean. You cannot fast for months, but you can do a very limited carnivore type diet for months, and then you will see the benefits. Because I'm certain if you have gut issues, you will see the benefits, and then you can reintroduce gradually, step by step, these higher fiber, so that the food component of this three-way relationship, you can reintroduce first the animal type of fiber, the collagens, the connective tissues, and then you can start adding the dairy, for example, which is you saw the casein provides feed for bacteria, and also the oligosaccharides in milk. And then you can start adding more complex plant foods and see what you react to and how you react to these food components. And when you react badly, you just eliminate that type of food. So I call this an elimination diet, and it works like a charm. So if you are ready to do something big, you go on a muscle meat only carnivore diet, and then add back gradually the connective tissues, and then the dairy products, and then the plant foods, and see what happens. What fits you, fits your microbes the best, or your microbes and the immune system best. Yeah, and one more interesting thing, it seems that dairy fat, not only the protein in dairy and the oligosaccharides, but the dairy fat has a big impact on the bacteria in the gut. So that also helps the infant, the breastfed infant, with the regulation of bacterial composition. It was tested in mice, but I think I have a similar study in other but also anecdotes from humans. When they go on a very high milk fat diet, a lot of their gut issues disappear. And that's because the high milk fat can regulate the bacteria in itself. This was called a very high fat diet, 45% by weight. I think 45% by weight is more than 70% by calories of fat, probably 80% fat, and that fat in this specific study was derived from milk fat. and that's very important. And what happened when they increased from 20 to 45? Again, it's not calories, so it's not a 45 calories, but something like I don't know, almost double. So from a 40% calorie fat diet, they went on a almost 90% calories milk fat diet. Lower endotoxemia occurred in parallel with an increased number of intestinal goblet cells. So somehow this milk fat stimulated the mucus producing cells, and the animal started releasing more mucus. I think it can also be a problem for people who have asthma and who deal with excess mucus. If they eat a lot of milk fat, I think that's another side of the same coin. If you have a different problem, the excess mucus produced can also be a problem. But for this scientific experiment, I think it was very nice to see that the milk fat also has a role in regulating the microbes. So the takeaways I added to the end. No, you are absolutely not eaten to disease or alive by microbes in a natural low fiber context. Natural means the context we discussed. If you are on a carnivore diet, if you fast, if you are breastfed, you will not be eaten. You will be healthier actually than what you had been before. And that context is very different when they use the mice and studied what happens on that high-ish fat, ultra-refined diet. That's a completely different context, and it shouldn't be interpreted or translated one-to-one to humans in a different context. And animal foods also contain fermentable fiber. We don't normally call it fiber, but actually if microbes ferment it to short-chain fatty acids and to some gas, then it's essentially and practically fiber. And it's especially rich in connective tissues. Evidence derived from proper context suggests that both connective tissues and your own mucus can maintain a balanced gut microbiota. Always follow the evidence and consider context. I think that's kind of advice I put at the end of any topic because biology is so heavily contextual that if you fail to consider context, you are just shouting stupid things in the room.
Fantastic stuff. Thank you, Gabor. So there's a couple of questions I have.
Sure.
I just want to be respectful of your time, though. Do you have time for a few questions?
Yeah, I think 5-10 minutes I can stay. Then I need to run for the kids.
Okay, no problem. So maybe you can stop your share and then we can.
Oh, sure.
Yeah. So the main takeaway, if I can put it in my own words, is that the digestive system is very complex. The microbiome is very complex. Not much is understood about it, but based on a plurality of data, there is no reason to assume, or there's no reason to conclude that fiber is an essential nutrient for the microbiome. That's what I'm taking away. Is that correct?
Yeah, I think we need to be pedantic. So we need to say that exogenous, so food-derived plant fiber, is not essential because you can substitute it with connective tissues, for example, in your
Yes.
food, or you can just go fasting or increase your milk fat content and increase your mucus production and feed your bacteria internally.
Yeah, and another takeaway, so related to that, it's so little understood that if we had someone say we had someone who'd been on a vegan diet for like 6 years and someone who'd been on a carnivorous diet, purely carnivorous diet for 6 years, there's no reason to conclude, or we definitely cannot even guess which of them may have. First of all, I don't know how to say which microbiome is better. The way we measure microbiome being better is with microbiome diversity. A, there's no way to tell which of those has the most diverse microbiome. And B, there's no way to know, even if one may be more diverse than the other, is that a good thing or a bad thing? Like we don't really know. Is all of that correct?
Yeah. I think microbiome diversity is expected to go up if you provide high amounts of fermentable fiber from exogenous sources. So whether it's copious amounts of connective tissue or fermentable plant fibers. This will help the your luminal microbes to proliferate. So, and that is what you measure when you take fecal samples, your excrements, and measure the bacteria. But you don't really see what happens at the surfaces, because as you saw from at least one study I presented, these are fairly fixed bacteria which adhere to the mucus, to the surface of the surface layer of the intestinal tract, and then uh, you don't really know what's happening. Is there any change? Is there a change for the better? And those are the most important things, because all the immune interactions, all the metabolic interactions occur at the surface. And then it's not even the colonic surface, the large intestinal surface, but 85-90% of the surface is in the small intestinal tract, which we even more rarely measure, or barely anytime it is measured. So we don't. There are some studies, and I have a compilation. I have a probably unusually high number of studies gathered over the years. But in general, if you see the explosion of studies on the microbiome, on your gut microbes, then the vast majority was done on fecal samples. So
Yeah.
we know very little, and even if there is some data, it is mostly derived from the colonic mucosa and not from the small intestinal, because it's simply difficult to access.
It's very difficult to access. And my understanding, this is from something I read maybe many years ago, so probably it's wrong. But my understanding was that because of the higher acidity, so the stomach has a certain acidity that which is very high, 1.5, 1.2 to 1.5, I think is the stomach acidity. Then the small intestine has, I think it's 2.5 to 3.5, and only the large intestine has a much lower acidity. I want to say 4 or 5. I'm not going to remember exactly. But my understanding was that because of the higher acidity, you actually can't have much of a microbiome in the small intestine. But that's incorrect. Is that correct?
I think it's technically correct if you go by the numbers, the number of bacteria or mass of bacteria, but it's not really what I believe matters. And also the pH changes very quickly when your stomach releases the foodstuff, the pancreatic excretions, the bicarbonate and other things quickly increase the pH to neutral levels. So it's only the uppermost part of the duodenum which faces this big range of pH values. But in fact, and also the transit speed is much higher in the small intestinal tract. It's just minutes or hours. It's not like
24 hours.
days, 1 or 2 days. If you eat a high plant fiber diet, then it can stay for 2 days and then fermenting. This is not what happens. So that's why the mucosa attached bacteria is the important ones, because we almost exclusively find those in the small intestinal tract, which are attached to this enormous surface and interact with the immune system and the hormone secreting cells and all the others. So they influence your metabolism in a much greater way than your colonic bacteria.
Right. And in terms of so I have clients who ask very specific questions, and I'll tell you how I answer the question, and then maybe you can tell me your answer, and that'll be our last question for today. The question is, should I take, very simple, should I take, there's a new bifidobacteria strain of supplement I can get, or lactobacillus, whatever it may be, or Akkermansia has become popular now, and there's a new company that has figured out a way to actually make Akkermansia in a lab, which is actually quite difficult, it turns out. So anyway, so the question is, should I buy the supplement or not? And my answer is, I don't know. As far as I can tell, there's no reason based on your symptoms to do it. But if you want and if you see an improvement in symptoms, then, it's a shot in the dark is basically my advice. What would you say?
I think there are several questions in this one, because one thing is taking a supplement, and another thing if it will colonize you, so it will stay, or you just taking the pills and then releasing it at the other end. Then it's kind of throwing the money out of the window. But also there is again context. So bifidobacteria, as I claimed, and I'm confident that I'm right, they play a big role in the infant and toddler microbiota. And when we transition to adolescents and then adults, our basic microbiome setup changes. And not only they are not dominant in the adult microbiota, they are not even as important. For example, a different context, when you are on a ketogenic or carnivore, so when you have high levels of ketones, these ketones will actively inhibit the growth of bifidobacteria. Is it a bad thing? There are many health benefits of ketogenic diet. You just search the literature, and even if it's not very popular among researchers because they cannot get funding from pharma, food, and from other places, there are still an abundance of studies which show different benefits of a ketogenic diet. But how can it be beneficial if it actively suppresses bifidobacteria, which are considered the really good guys? These are not necessarily the really good guys. It depends on context. Again, in a ketogenic context, they are not needed. They are actively suppressed by the ketones.
It's fascinating. So there's a number of questions. One, do we even know, given your specific, given my client specific, the client who's asking, their specific context, do we even know what would be a good microbe to add to the context? I think my short answer is no. Number two is, will that actually stick? Like, how do you know that you're not just going to poop it out? How do you know it's not going to just, how will it survive the high stomach acidity, in the first part of the digestive tract? There are all these questions that are not clear. I will say anecdotally, some people claim to get benefits, they claim that they took a pill and it stopped certain symptoms and so on. So anecdotally, there's there, but there's also placebo effects, so that doesn't mean much. But I mean, what I'm hearing is it's a shot in the dark. Like many other interventions, we have no idea.
You just don't know what you are doing. And if anybody tells you the opposite, that this strain of whatever bacteria were clearly shown that it works in everybody in the same way, that's probably not true. At least you should be eating or feeding symbiotics, so some of the probiotic bacteria and some of the prebiotic feed for these bacteria. And when you eat sauerkraut or kefir or these kind of fermented foods, you actually do that. So I'm much more a fan of if you believe in fermented foods, and it's very easy because you try, and if it works, you feel better, then eat fermented foods. That's very easy. It's just eating plain bacteria for some purported benefit. I think is really shooting in the dark. And there is I remember that there is a pair of studies also in Cell published in 2018 when it was actually shown that some of these supplemental bacterial probiotics can actually do harm.
I have no doubt about that last point, and I think that's a good point to leave it. Gabor Erdosi, it's been a real pleasure. I hope we can do this again sometime. Thank you for your time.
Yeah, you're welcome. It was a pleasure.
Thanks for listening to my conversation with Gabor Erdosi. For more content like this, check out my website, samirdosani.net. Thanks, and I'll see you in the next one.